Phase transfer catalyst method is relatively a new technique for the detection of latent fingerprints on crime scene evidence. The composition is prepared by mixing equimolar solutions of a xanthine dye and a phase transfer catalyst and preparing them in water. Normally, tertiary tetrabutyl ammonium iodide is used as the catalyst. As far as the dyes are concerned, from the xanthine classification, eosine yellow, eosine blue, erythrocene and rose bengal are believed to give the best results. The composition basically tags the insoluble lipid and protein content of latent fingerprint residue and develops sharp and clear prints on a wide number of surfaces. This is the beauty of the method, that it has got broad-based applications. It detects fingerprints on porous items like paper and cardboard, on semi-porous items like currency notes and glossy paper, as well as on non-porous items like metals and plastics. Since the lipid and the protein contents, which is tags, are insoluble in water, therefore, recently it has been found out that this method may be extended for detection of latent fingerprints on those surfaces which the criminal has either accidentally or deliberately washed after committing the crime. We have seen that those items which have remained immersed in water for almost 24 hours also give beautiful fingerprints by this method. The composition is in form of spray, but since water is the medium, this is not much of a problem. Water is not only easily available, but as compared to organic solvents, it is always safe to use. The organic solvents like benzene or ether are either inflammable or they are explosive in nature. No such problem is encountered when water is the medium. Moreover, these days people normally use ball pen or gel pen for writing. And suppose fingerprints are to be detected on any document, then by spraying water, the calligraphic script is not despoiled. On the other hand, if some organic solvent is used, there is always a threat that the ink will run and the writing will be spoiled. Phase transfer catalyst method takes just 2 minutes to detect fingerprints. It's cost effective, no specialized equipment, no costly instrument is required for this method. The technique is very easy to operate and does not require any specialized handling. And given the fact that such large number of crime scene evidence may be processed by it, this technique is useful since it is not specific of surface as well. So, when it is given to the police officers, they need not apply their mind as to which composition has to be used for which surface, because this will act on almost all types of crime scene evidence which are generally encountered at scene of crime.
Dear students, after studying this module, you shall be able to know that a combination of a phase transfer catalyst and a xanthine dye acts as a fingerprint reagent. Along with it, you'll learn that the phase transfer catalyst method detects fingerprints on both absorbent and non-absorbent crime scene evidence. It is possible to detect fingerprints on a wide variety of adhesive tapes by this method, and you'll learn that it is possible to detect fingerprints on wet items by this method. You see, a latent fingerprint is formed when the sweat pores of the papillary ridges leave a deposition of perspiration on a surface with which the finger has been brought into contact. Deliberate impressions of fingerprints may be formed by ink or other substances transferred from the peak of friction ridges on the skin to a relatively smooth surface such as fingerprint card. Fingerprint records normally contain impressions from the pad on the last joint of fingers and thumbs, although fingerprint cards also typically record portions of lower joint areas on the fingers. Human fingerprints are detailed, presumed to be nearly unique, difficult to alter, and durable over the life of an individual, making them suitable as long term markers of human identity. They may be employed by police or other authorities to identify individuals who wish to conceal their identity or to identify people who are incapacitated or deceased and thus unable to identify themselves, as in the aftermath of the natural disaster. Human body possesses the following three types of glands, that are eccrine, apocrine, and sebaceous, the secretion of which contribute to a fingerprint deposit. Let us study eccrine glands in detail. Eccrine glands are widely distributed throughout the body and are particularly numerous on the palms of hands and the soles of feet. These glands secrete chemicals as a result of general metabolism and catabolism. They reach their peak of development in humans, where they may number 200 to 400 per square centimeter of skin surface. They produce a clear, odorless substance consisting primarily of water and NaCl. NaCl is reabsorbed in the duct to reduce salt loss, but is dysfunctional in cystic fibrosis, thus producing salty sweat. They are active in thermoregulation by providing cooling from water evaporation of sweat secreted by the glands on the body surface and emotional induced sweating. Besides water content, eccrine sweat contains up to 1% of the other substances, of which inorganic salts constitute about one half. The other half is constituted by organic products like urea, creatinine, choline, lactic acid, sugars, uric acid, amino acids, and proteins. The constituents of sweat may be selectively fixed by different chemical reagents so as to make the latent fingerprint visible. One of the recent methods of fingerprint detection involves the use of phase transfer catalyst in concert with a xanthan dye. The commonly employed phase transfer catalyst is T-tetrabutyl ammonium iodide. The following dyes belonging to the xanthanin classification may be used to prepare the fingerprint formulation eosin Y, eosin B, floxin B, erythrocin B, or rose bengal. Now let us discuss various eosins in detail. Eosin Y. Eosin Y is a form of eosin. It is most commonly used as an acidic red stain for highlighting cytoplasm material in samples. It has a very slightly yellowish cast. Then comes eosin B. The other eosin compound is eosin B. It has a very faint bluish cast. Floxin B. Floxin B commonly known simply as floxin, is a red colored dye that is used in the hematoxylin floxin saffron stain. Rose Bengal. Rose Bengal is a stain. Its sodium salt is commonly used in eye drops to stain damaged conjunctival and corneal cells and thereby identify damage to the eye. The quality of developed prints is comparable to other chemical development methods. Then comes mechanism by which phase transfer catalyst works. The weak or no development of latent prints may be due to the difference in phases of reagent and the residue. PTC is a heterogeneous catalysis where a catalyst facilitates the migration of a reactant from one phase to another. It comprises of a hydrophilic phase, a hydrophobic phase, and an interphase. The ion gets transferred from the aqueous to the organic phase. T-tetra-butyl ammonium chloride and T-tetra-butyl ammonium iodide have been reported as efficient phase transfer catalysts. It involves formation of a metal carbanion at the interface. The method is efficient for both porous and non-porous surfaces. Tetrabutyl ammonium iodide has been incorporated as the precipitating agent to accelerate the reaction between the insoluble calcium ions of the fingerprint residue and an aqueous solution of disodium salt of eosin. Workers have successfully developed latent prints on the sticky side of adhesive tapes using PTC method. Now let us see the methodology. Latent fingerprints could be developed very effectively with the help of a very simple method using phase transfer catalyst. The reagent is inexpensive, non-toxic and easy to prepare. The reagent is equally effective in cases of fresh as well as older prints. The PTC and Rose Bengal dye formulation develops fingerprints on all types of adhesive tapes of such a quality that even third level detail can be seen. The formulation is prepared by dissolving a mixture of T-tetrabutyl ammonium iodide and any one of the aforementioned xanthan dye in minimum quantity of water. The contents are stirred for about 30 minutes. Small pieces of paper, lamination sheet, gift paper, adhesive tape, or polythene bearing sample latent fingerprints are dipped in the test solution for about 1 minute. The paper samples are then immersed in distilled water for about 30 seconds. Other surfaces are dried with a hair dryer. For larger objects such as polythene bottles, bakelite switches, glassware, and steel utensils, the test solution is sprayed onto the area containing the fingerprint impression. The surface is then dried with a hair dryer. Now let us have a look on the results. The phase transfer method of fingerprint detection offers the following advantages. Number 1, the reagent used are cost effective and non-toxic. Number 2, the test solution is prepared in water. In ninhydrin method, the solution is prepared in organic solvents which are invariably inflammable and or explosive. Moreover, when these solvents are sprayed over a document, the calligraphic script gets despoiled. Third, the formulation develops sharp and clear fingerprints. Fourth, it takes just 2 minutes to develop latent impressions. In contrast, ninhydrin and cyanoacrylate methods take 1 to 2 hours to develop fingerprints. Fifth, the method has broad-based application. It detects fingerprints on a wide spectrum of objects, absorbent and non-absorbent, white and colored, and smooth and rough. Sixth, the formulations develop fingerprints on a wide variety of adhesive tapes, including doctor's tape, packaging tape, cello tape, and duct tape. A representative example in the given figure. It is possible to develop fingerprints on wet items with the aid of such formulations. A sample fingerprint developed on a spoon which had remained immersed in water for about 12 hours. 8. The technique does not require costly equipment or a sophisticated instrument and hence is suitable for a developing country like India. Dear students, now let us summarize this chapter. A latent fingerprint is formed when the sweat pores of the papillary ridges leave a deposition of perspiration on a surface with which the finger has been brought into contact. That human fingerprints are detailed, presumed to be nearly unique, difficult to alter, and durable over the life of an individual, making them suitable as long term markers of human identity. That eccrine glands are widely distributed throughout the body and are particularly numerous on the palms of hands and the soles of the feet. We have also come to know that the phase transfer method is a relatively novel technique of detecting fingerprints on crime scene evidence. Its advantage is that it is not surface specific, that is, it can develop impressions on a very large number of articles of divergent colors, textures, and constitutions. Also develops fingerprints on moist and sticky items. The method is cost effective and does not pose an occupational hazard to the user. PTC is a heterogeneous catalysis where a catalyst facilitates the migration of a reactant from one phase to another, and it comprises of a hydrophilic phase, a hydrophobic phase, and an interphase. Method is efficient for both porous and non-porous surfaces. And lastly, for larger objects such as polythene bottles, bakelite switches, glassware, and steel utensils, the test solution is sprayed onto the area containing the fingerprint impression.